I am Sean Mayfield, and I did my project on carbon quantum dots. So, carbon quantum dots are nanoparticles that are typically less than 10 nanometers across, but they can be found to be as wide as 15 nanometers across, which means they're made of mostly carbon and can range from about 16 to 80 carbon molecules or carbon atoms. And they were first synthesized in 2004 by accident on a study in the tensile strength of carbon nanotubes, but were later studied and identified in 2006. So one of the main properties of them is functionalization, which is the ability to attach different functional groups to the huge amount of hydrogen atoms on the outside, which creates a variety of actual properties, and they differ from regular quantum dots in structure and makeup, as normal quantum dots are made of silicate and metals, whereas these are made of mostly carbon and other organic molecules. So the two key properties to a carbon quantum dot is range in their optical properties and the fact that they emit fluorescent light or different wavelengths of UV light under fluorescent light. And the wavelengths depend on the size of the molecule and what functional groups or atoms are attached, like in the picture on the right. They have different things like nitrogen, oxygen, sulfur, and hydrogen, and then also in size on the left, the different sizes of them, the smallest emitting the largest wavelengths or lowest energy wavelengths of light, right? Light on the right side, and then the larger ones emit smaller wavelengths of light. And then another one that isn't really a property but creates a lot of the different properties of these is functionalization, which was previously mentioned. So with all of these different properties come a variety of applications. First one being bioimaging, which is just taking pictures of small things, which because these carbon quantum dots uh, can emit different wavelengths depending on what's interacting with them, makes them incredibly practical for this. Another application is used in drug delivery. Now, they wouldn't be used as any sort of drugs or medicines themselves, but because they can attach these functional groups, it makes them very useful for delivering high volumes of small drugs into the body with very little molecular application. And then they also work as photocatalysts because of the wavelengths of light they emit being incredibly precise. And then there was another study in 2018 that suggested that they were used for desalination of salt water and purification in that because of citric acid created ones shown on the bottom. And then they're used in solar cells again, depending on their optical properties, electronics and chemical sensors. They have a whole bunch of applications depending on what groups they're attached, and the size of these molecules. So, uh, with the variety of applications, it seems pretty obvious what the benefits of these are. So I'm jumping right into the drawbacks. And the drawbacks do depend on the application. For instance, in biomedical applications, the fact that they don't themselves easily diffuse across the cell membrane means it's incredibly difficult for them to get into the cell. But white blood cells were found to be breaking them down to make it easier to cross certain barriers, like the blood-brain barrier, in a study in 2016, which suggested they could use to treat Alzheimer's, and you could attach different solvents as functional groups to these molecules, and then the solvents would break down and diffuse across the blood-brain barrier much more easily than traditionally delivering them just into the blood with other delivery systems. Another problem with them is that they have incredibly long decomposition rates, which is a disadvantage for a biomedical application. But for the previously mentioned solar cell application, that was actually one of the main benefits towards using them in there. That in the type of solar cells they were used, it was a dye-controlled solar cell, which changed color depending on the type of light that they're getting from the sun. But the long decomposition rate was one of the main selling points in that application. So drawbacks do depend on the application, as they could be a problem in one application but a huge advantage in another application. And then there is one major drawback in all situations is that it's very difficult to 100% purify many types of these from other carbon quantum dots, and it's very difficult to control the sizes down to a very fine point. And these are just different methods of creating carbon quantum dots on the right and showing the different yields of them, and then which leads into the synthesis of these carbon quantum dots. And there are two main types of carbon quantum dot synthesis. One of them is the top-down method, which is breaking down larger materials through microwave irradiation, chemical oxidation, and laser ablation, which they take large sheets of typically graphene and introduce purposeful impurities, which is typically nitric acid and water. And then once these impurities spread out to an even size, they consistently can cut down with the irradiation the graphene into regularly shaped carbon quantum dots. And then there's bottoms up methods, which is synthesis from smaller molecules such as carbohydrates, polymers, and citric acid, which was used in the 2016 study on desalination, previously mentioned. So I'll show what my model looks like in this picture. This is just a graphene quantum dot here, which it's made of just the regular shapes with no functional groups attached. And carbon quantum dots can be one layer of carbon atoms essentially, or it can be many, typically three. And this is my model.